So, osteosarcoma, most common bone tumor of childhood. It is a disease that metastasizes early, and certainly about 1/5 of the patients have metastatic disease when they first present. Chemotherapy has some benefit, but it's not dramatic. And if you can't cut this thing out, you've got big trouble with the disease. And so if a patient happens to have a sarcoma that doesn't start, an osteo that doesn't start in a place where it's easy to operate on, then those patients typically have a very hard road to hoe. So we've got a lot of work to do in sarcoma. The last major advance came in the late 80s when we learned that adding chemo helped. And so it's, again, a lot like the other pediatric diseases. We're kind of still, in the dark ages, I think. Genetically, it's a very, it has something called chromothripsis, which means their genes are completely in disarray. There is no one chromosomal mutation that can be targeted. So it's really a tough challenge. And for these kind of diseases, the immunotherapy that the Nobel was awarded to today, even though this has had incredible benefit for things like melanoma and lung cancer, it's had essentially no benefit for osteosarcoma. We've tried it in osteosarcoma and it doesn't work. It hasn't worked in most sarcomas. So we got to do something else. And we've been trying to develop chimeric antigen receptor-based gene therapy for osteosarcoma. We know that it expresses a target that we can go after with CAR T cells. We know that we can make CAR T cells that go after the target. But what happened was that we started a clinical trial back in about 2011 using these CAR T cells, and it didn't work very well. And the more we studied the CAR that we had made to target osteosarcoma, the more we learned it was defective. It was just a defective CAR. We didn't know it at the time, but it is a problem with the receptor. It clumps on itself, it aggregates, and it causes a signal in the T cells that causes them to become dysfunctional. So we've spent the better part of the last 5 years trying to fix this CAR. And we've been going about it many different ways to try to reduce the clumping or to try to reduce the exhaustion that results from the clumping. And we've got now several approaches that we think are going to render it more potent when we go into the clinic. And so we finally now, after all of this re-engineering and reconfiguring, we're ready to go back into the clinic. Now, when we do it this time, though, we don't want to do it just as a single agent study. We want to use the CAR T cells, and we also want to give a drug to modulate the immunosuppression that we also know is happening in these patients. So what the funding from ACGD has done, and Wendy's Walk and Swim Across America, has really allowed us to go back and look at the CAR from before and say, why didn't it work? How can we reconfigure this treatment regimen to make it more effective? And we're, hoping to launch this trial in, fairly early in 2019.